I'm Nikki Dix. I'm the research coordinator at the Guana, Tolomato, Matanzas, NERRS in St. Augustine, and I'm the project lead and collaborative lead for this project. Also at our reserve and on this team is Shannon Dunnigan, who's our system-wide monitoring program manager, and she's serving as the data manager for everything. And then Silas is our swamp technician, and he's one of the co-technical leads on the project. We also have the other co-technical lead is Eric Smith at North Inlet, used to be a research coordinator, now a manager. And then we have 11 other reserves with research staff who are also really interested in monitoring chlorophyll. Next slide. So why chlorophyll? It's hard for me to back up and talk this broad sometimes, but chlorophyll is basically the most practical way that we can figure out how much phytoplankton's in the water. And the amount of phytoplankton in the water, it tells us a lot about the health of the ecosystem. So this is a good conceptual diagram that Dr. Bricker created. Next slide. So currently our standardized national monitoring program requires us to collect monthly samples for chlorophyll. So we go out to the site, we collect some volume of water, we filter that water down to concentrate all the plankton on a filter. And then you put the filter and some acetone to extract all of the chlorophyll pigment into solution. And then you put it on a machine like the one here. It's a fluorometer and it measures the fluorescence of chlorophyll. So all the reserves at every site have some time series of monthly chlorophyll data that looks something like this, which is good. We can track long-term trends and look at seasonal patterns. The problem is that plankton live and die on the scale of about days. And also at a particular site in an estuary, the water is moving around so much, the plankton concentration in the water can be really variable. So monthly measurements are missing a lot. And we're also almost certainly missing some algae blooms, which can be a public health concern. Next slide. So at all our sites, we also have these YSI data loggers getting 15-minute measurements on things like salinity, temperature, dissolved oxygen. And a few years ago, YSI developed a new sensor that measures chlorophyll. So the benefits of this high frequency chlorophyll, like the graph you see in the middle, is that you can look at this really short-term changes in chlorophyll and get those blooms. A few reserves had purchased chlorophyll sensors. They're really expensive though. They're about $3,500 a piece. And so they've started, testing them out, and some of the data looks really comparable to the monthly grab sample data, but some of it doesn't. Also, sometimes they see spikes in chlorophyll from the sensors, and we're not sure if these are real increases in algae or if there's some kind of interference in the sensor. And I love how that picture got turned sideways. I was going to say that one of the benefits of these high frequency data also are that there's the potential that they can be sent up to the satellite and delivered real time, just like our other 15-minute data. Next slide. So all 13 reserves in this project will be taking side-by-side measurements of chlorophyll using the two different techniques to compare them. And we'll be generating lots of these comparison graphs. We'll generate a graph like this and quantify the relationship between the sensor chlorophyll and the extracted chlorophyll for every site. And then we're also look at making a graph like this in a relationship for all the sites combined. We'll do lab experiments to look at sensor interferences and develop corrections. And then we'll develop recommendations for the NERRS monitoring program. The NERRS staff really are our direct end users, but we're also engaging stakeholders who monitor chlorophyll, like state agencies, as well as educators at all of our reserves, because they teach about a lot of these concepts. Next slide. So we only have a year to do all of this. We have project team meetings every month. They've been going really well. Our comparisons and interference tests will go through the summer. And then at the end, we hope to have a writing workshop, probably virtual, make recommendations, and then hopefully have a system-wide webinar as well as other products.